Welcome back everyone. I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about genetic cross in Drosophila, and we will be mainly focusing on 100% male lethality or double lethality in Drosophila Carli mutation. Okay. So in this series I have already made 4 videos. In the first 3 videos I have discussed about eye color mutation, vestigial wing mutation and Carli wing mutation in Drosophila. And in my fourth video, I have discussed about 50% male lethality in case of Carli mutation in Drosophila. So if you want to watch those videos, I will give the link below in the description box, and I will also attach it at the end of this video. Okay, you can watch it out. So before beginning the video, I would like to request you to kindly subscribe to my channel for more such videos, and if you find this video useful, don't forget to hit the like button and share it with your friends. Okay, so without further delay, let's begin. So first of all, what is Carli mutation? See, in my previous 2 videos of Carli mutation, I have already discussed in details. Okay, so here I am only briefing it. See, Carli is a wing mutation in Drosophila, where you can see if Carli mutant alleles are there, you can find a backwardly curled up wing in case of Drosophila. Okay, so Carli mutant allele is denoted as CY, and you have to remember that phenotypically this mutant allele is dominant over the wild type of wing allele or normal wing allele, that is denoted as CY plus. Okay, so CY is dominant over CY plus phenotypically. Okay, and it is autosomal dominant because the allele is present on the second chromosome, which is an autosome of Drosophila. Okay. And another thing, you must remember that CY is also a recessive lethal allele. Okay. So what is the importance of recessive lethal allele here? See here, we are seeing that CY is dominant over CY plus. So obviously in both homozygous and heterozygous condition of CY, it should show Carli wing phenotype. Okay. But you can see in case of heterozygous condition where one chromosome contains CY and another contains CY plus, it's showing Carli wing. Okay. But in case of homozygous condition, when both the chromosomes are both the chromosomes have CY, it is not showing Carli wing. Rather, the progeny is here lethal. Okay, they does not survive. Why? See here, because CY or Carli is a recessive lethal allele. That means whenever it will be present in homozygous condition, the progeny could not survive. It will get dead because it is a recessive lethal allele. Okay. So only though it is dominant over CY plus, it can only show its phenotype in heterozygous condition but not in homozygous condition. Okay. And as the CY plus is recessive to CY, so obviously in homozygous condition only CY plus will show its phenotype, that is the wild type wing. Okay. So this is all about Carli mutation. Now 100% male lethality or double lethality means there will be no male progeny survived. Okay. So let's check it out through an example. See here, the question says this is the data of F1 progeny. Okay, where you can see only females are there, Carli winged females and wild type females. Okay, and their respective numbers are given, and you have to derive the P generation cross from here. Okay. So before going into details, first of all we have to derive the sex ratio here. The sex ratio male is to female equal to 0:30. That means there is no male present here or 100% male is absent. Okay. It is very important. When 100% male is absent, how will you derive the P generation cross? Next, the phenotypic ratio is Carli is to wild equal to 20:10, that is 2:1. We know that 2:1 is the modified monohybrid cross ratio when recessive lethal is present. Okay, we generally know 3:1 is the monohybrid cross ratio, but in presence of recessive lethal allele like Carli allele, the ratio becomes 2:1. Okay. Now from our previous videos we have learned that whenever there is a monohybrid cross ratio, we have to take both the parents as heterozygotes. Okay. So here in the P generation cross, you can see I have taken mother as heterozygous for the CY allele. Okay. CY plus CY I have taken here, and I have also taken father as heterozygous, okay, CY plus CY, okay. That is fair enough for phenotypic ratio. Now you have to introduce the 100% male lethality here also, okay. See here, in case of 100% male lethality, in case of 50% male lethality, we have considered one lethal allele on X chromosome, okay. But in case of 100% male lethality, one lethal allele, that is L, is not sufficient. We have to take two lethal alleles, that is L1 and L2. Okay, got it? L1 and L2. And you have to remember this, these two mutant form of lethal alleles, okay, are recessive to their wild form, that is L1 plus and L2 plus, okay. L1 plus and L2 plus. So L1 plus and L2 plus are the wild type forms of these lethal alleles, and L1, L2 are their mutant form. When L1 or L2 is present on any chromosome, that means the product of that allele L1 or L2 cannot be formed. So the progeny may get dead if their complementary allele is not present. Okay, what is that? See here. First of all, I have taken the two X chromosome of mother. One is with L1 plus and L2. So here L2 is the mutant form of the lethal allele, and on the other homologue I have taken L1 and L2 plus. So here L1 has the mutant form. So obviously, why this mother is viable, though there are two lethal alleles, mutant form of lethal alleles are present here? Because though here is L2 on this chromosome, but its complementary allele L2 plus is present on its homologue. Okay, that is how this L2 plus is complementing or is providing the protein that is not produced by this L2 mutant allele. Okay. On the other hand, though there is L1 that cannot produce protein, but its complementary wild type allele, that is L1 plus, is present here, which is complementing the absence of protein formed by L1 from this chromosome. Okay, so that is how these two are complementing each other and the mother is viable, though it has two mutant alleles. Okay. But in case of father, you can see I have taken both the lethal allele in its wild type form. Why? Because if one is in its mutant form, say here is L1, that means there will be no complementing allele for L1. Okay, because L1 cannot produce any protein, the mutant allele cannot produce any protein, and male have only one X chromosome. So to complement the absence of L1 in one X chromosome, there will be no L1 plus in another X chromosome. Okay, because male have only one X chromosome. So for that we have to take both the lethal alleles in its wild type form for male, either or the male cannot survive. Okay, got it? Now we will see how taking lethal alleles in heterozygous condition for female we can derive all male progeny as lethal. Okay, see here, when this X chromosome will combine with this Y chromosome of father, it will form L1 plus L2 and Y chromosome. So obviously the product of L2 will not be there, so the progeny will get dead. Got it? When this X chromosome of mother will combine with Y chromosome of father, it will not produce the product of L1. Okay, so obviously here also the progeny will get dead. Male progeny will be dead. Okay. On the other hand, let's take another color from here. When this X chromosome of mother will combine with this X chromosome of father, though it does not have L2 here on mother chromosome, but L2 plus is here. Okay, so product of L2 and L1 will be formed after combination of both the X chromosome from father and mother. So it will be viable female individual. Okay. On the other hand, when this X chromosome will combine with this X chromosome from father, product of L1 will be from father side and product of L2 will be from both the father and mother side. So this one will also be a viable female. Okay, got it. Now another thing you must see here that is very important here to notice is that I have taken an I here on the X chromosome of mother. What is this I? This I is the inhibitor of recombination. See, L1 and L2 alleles are present on the same chromosome. So there is a chance of recombination between these two loci. Okay. So if recombination occurs, what will happen? One X chromosome will be L1 plus L2 plus. Okay. So one X chromosome will contain L1 plus and L2 plus, while the other one will contain L1 and L2. Okay. L1 and L2. See here, when these L1 and L2 will combine with the Y chromosome of father, the progeny will not survive. But when the L1 plus L2 plus will combine with Y chromosome of father, then the progeny will survive. So the male progeny will be survived in the F1 generation. But in our question, we have seen that no male progeny is found in the F1 generation. So if recombination occurs, then male progeny will survive due to this recombinant X chromosome from mother, and that could not be possible here. That is why, to stop the formation of this type of recombinant X chromosome from mother, we have to apply an inhibitor of recombination here on the X chromosome of mother. Got it? So I think now why we have taken this type of cross in case of father and mother in the parental generation is now clear to you. Okay. If you have any doubts, you can give a comment below in the comment section. So now let's dig into deep. Okay. So see here, from the mother we can find four types of gametes. One will be CY plus and this X chromosome, that is L1 plus L2. Another one will be CY plus and this X chromosome, that is L1 L2 plus. Then CY and this X chromosome, that is CY and L1 plus L2, and another one will be CY with this X chromosome, that is CY L1 L2 plus. Okay. From the father we can also get four types of gametes like CY plus and X chromosome. See here, CY plus and X chromosome. Then CY plus and Y chromosome, CY plus and Y chromosome. Then CY and X chromosome, CY and X chromosome, and CY and Y chromosome, CY and Y chromosome. Okay. When we arrange all these alleles from father and mother, okay, we can see 16 progenies are formed. Okay. But you can see here many of them are lethal. So let's see how their phenotypes can be determined. See here, first it is CY plus, that means it will be wild. Okay. It has two X chromosomes, so it will be a female. And see here, though there is a lack of L2 plus allele on this, but it is complemented by L2 plus allele on its other homologue. Okay, so it will be a surviving wild female. Then CY plus, so it is obviously wild, and 2X chromosome, that means it's female, and here wild type allele of L1 and L2 both are present, so it will be surviving. Then CY plus CY, so it will be Carly, okay, and it has 2X chromosomes, so it will be female, and as it has both wild form of L1 and L2, so it will be a surviving female. Then CY plus CY, so it is Carly, and it has 2X chromosome with both wild form of L1 and L2, so it will be a surviving Carly female. Okay. Then CY plus CY, so it is wild type, but see here there is only X and Y chromosome, and the only X chromosome contains L1 plus allele but no L2 plus allele, so it will be lethal. Okay, due to absence of L2 plus allele, it will be a lethal male. Okay. Then CY plus CY with X chromosome, Y chromosome, but X chromosome does not have the lethal have the wild type allele for L1, so it will be a lethal male. Okay. Then CY plus with X and Y chromosome, but X chromosome lacks the wild type allele for L2, so it will be a lethal male. CY plus with X and Y chromosome. X chromosome does not have the wild type allele of L1, so it will be a lethal male. Okay, so you can see 4 males we have seen here, and 4 are dead till now. Okay, then CY plus, so Carly, 2X chromosome, so female, and you can see wild type alleles of both L1 and L2 are here, so it will be a surviving Carly female. CY plus Carly, 2X chromosomes female, wild type allele of both L1 and L2 can be found, so it will be a surviving female. CY with 2X chromosome. See here, as the CY allele is present in homozygous condition here, and we know that CY is a recessive lethal allele, so this female will be lethal. Okay, so see it is a lethal female. Okay, so first time we are accounting a lethal female due to lethality in CY. Okay, next, same case here, CY, so this female is also lethal here. Okay, so females, 2 females are lethal here only due to presence of recessive lethal allele CY, not due to presence of lethal alleles L1, L2. Okay, got it. Then next, CY plus, but X and Y, here X lacks L2 plus allele, so it will be a lethal male. Then CY plus, but the X chromosome lacks L1 wild type allele, so it is also a lethal male. Then CY, so this male is already lethal due to presence of CY here, and this male is also lethal due to presence of CY here. Okay, so you can see here, among 16 progenies, how many are lethal? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Out of 16, 10 are lethal. Among them, 2 are female. Okay, not male, sorry. 2 are female and 8 are male. Okay, and these 2 females are lethal due to recessive lethal allele CY, not due to L1 or L2 allele. Okay, so 6 survived, and among these 6 surviving progenies, you can see there are 4 Carly progenies. This one, then you can see here another one, and you can see here another one. 4 Carly females are there and 2 wild females are there. See, 2 wild females are there. So among these 6, there are 4 Carly females, 2 wild type females. Okay, so the ratio is 4:2, that is 2:1. We can see here from the question over here that the Carly female is to wild male equal to 20:10. So it is also, the ratio is 2:1, which is matching with our derived ratio 2:1 from here. So obviously our cross is correct and it is proved. Okay. So this is all about the 100% male lethality. So in case of 100% male lethality, you have to take 2 lethal alleles L1 and L2, and you have to put both the L1 and L2 as in heterozygous form on mother. Okay, because if you put it on father, the mutant alleles, then father will itself get dead. Okay, so the cross will not be possible. So if you have to put the mutant alleles on mother in heterozygous form, so that the absence of wild type allele on one X chromosome can be complemented by presence of the wild type allele of that lethal type on another X chromosome. Okay, and that is how you could do the crosses here. So I hope the video is clear to you. If you have any doubt, you can comment down below. Okay, and I will answer that. And if you like this video, don't forget to hit the like button and share it with your friends, and don't forget to subscribe to my channel for more such videos. Thanks for watching and happy learning.